This video we explain about natural killer T cell. Here's the outline of this video. Natural killer T cells are a type of T cell that also has properties as an NK cell. NKT cells were discovered in 1986 and are considered the fourth type of lymphocyte after T cells, B cells, and NK cells. NKT cells make up about half of the T cell population in the liver and bone marrow. In the peripheral blood, however, they represent only 0.1% of T cells. The structure of an NKT cell looks like this. Helper T cells as well as antigen-presenting cells express many molecules on the plasma membrane, but NKT cells express only one type of T cell receptor, and this receptor is also characterized by a lack of diversity. Other cells have some types of receptors, but NKT cells have only one TCR receptor. Next, we will explain how NKT cells matured. 80% of NKT cells are made from pre-T cells. They differentiate before being DP stage. They exist in liver and they are essential for biological defense. The others differentiate from DP stage. They exist in lung, bone marrow, lymph node, and gastrointestinal tract. So how are T cells activated? The mechanism by which NKT cells are activated is this. NKT cells are activated via glycolipids in the CD1D of dendritic cells and the TCR of NKT cells. CD1 is a membrane protein on antigen-presenting cells, such as dendritic cells and B cells that presents non-peptidic antigens. Among them, CD1D is responsible for presenting glycolipids to NKT cells. NKT cells can induce cell death against cancer and other cells due to their NK cell properties, but their direct cytotoxic activity is limited due to the small amount present in the body. On the other hand, NKT cells have the ability to produce a large amount of cytokines. The production of helper T1 type cytokines, such as interferon gamma, activates the innate immune system, such as NK cells, and the acquired immune system, such as cytotoxic T cells. The production of Th2 type cytokines, such as IL-4, promotes antibody production by B cells and induces allergies. NKT cells are thought to be involved in the induction of allergic inflammation and in the suppression of non-allergic diseases by producing IL-17. Since about 90% of NKT cells are secondary characters in infection defense, but can produce large amounts of cytokines immediately without clonal proliferation, it is now believed that their in vivo role is to activate other immune cells. In other words, by activating other immune cells, they play a central role in inducing long-term immune memory that causes an anti-tumor effect over a long period of time. Here is the summary of this video. NKT cells receive antigen presentation from dendritic cells via CD1D molecules and recognize glycolipids. Then secrete cytokines and activate NK cells, T cells, and B cells to induce long-term immune memory.